Okay, so I'll go ahead and introduce. Dr. Pinckney is the director of Belle Baruch Institute for Marine and Coastal Sciences and a faculty member of the Department of Biological Sciences and Marine Science program at the University of South Carolina. So his general areas of interest include microbial ecology, microalgae, phytoplankton nutrient interactions and harmful algal blooms, and ecosystem eutrophication and estuarine and coastal habitats. Most of his work over the past 25 years has emphasized investigations of the ecophysiology of benthic and phytoplanktonic communities and their contribution to ecosystem functions. So without further ado, Dr. Pinckney, you may go ahead and begin.
Okay, thank you very much for the opportunity to come speak today. Am I loud enough for you to hear me?
Yep, you're good.
Okay, so I apologize that I can't be there today. It's some very interesting talks, but I'll do the best that I can remotely. I want to recognize the second author, Kaylin Gildy, who was a undergraduate student who was working on her senior thesis and did most of the real groundwork for this project. And she's the one that came to me and said, I'd like to look at some of the potential effects of the horizon oil spill on phytoplankton communities. So we designed a little project, and I'm going to tell you about the results of that project today. As we all know, the ecological impacts of crude oil on phytoplankton have been studied, but there have been very few studies that have looked at natural communities in situ, and especially for salt marsh estuaries. And a lot of these past studies have focused on single algal cultures in the laboratory, and that makes it very difficult to look at sublethal effects because you don't really have anything to compare it to. And cultured algae are by nature pretty happy phytoplankton because the conditions for them to grow have to be very conducive to their growth. And in nature, all phytoplankton are not necessarily happy in their environment. The other problem is that lab conditions can alter the toxicity somewhat, and a lot of these previous studies have focused on the water accommodated fraction, which is that part that dissolves in water. We wanted to look at the effects of the crude oil and let the water accommodated fraction occur in a natural manner rather than us forcing it and removing the potential toxic effects of the oil itself. So the idea, and the literature support this, is that crude is actually more toxic than the water accommodated fraction alone. So the objective was to look at short-term responses of these natural phytoplankton communities to exposures to 3 different levels of crude oil, 10 to 100 parts per million, 10, 50, and 100 parts per million, or microliters per liter. And the hypothesis that we're looking at was, do exposures to low concentrations have significant effects on that phytoplankton community in terms of its biomass, but also in terms of who is affected in that natural community by exposure to that oil. So that's sort of our hypothesis. And we did our study at North Inlet Estuary, which is located in coastal South Carolina. It's a high salinity estuary that receives very little freshwater input from Winyah Bay. Most of the exchange occurs through the inlet mouth, and so we have pretty high salinities there. The water was collected at a place known as Clambank Landing, so that the communities were mostly phytoplanktonic and not benthic resuspended species. So we did our collections at high tide and high salinities. The experimental design was to collect the water from the estuary, We were limited in time. We only did it in February and March, which are the winter time. Water temperatures are relatively cool, and total phytoplankton biomass is not very high during that time of the year. We were able to get samples of 2 different types of crude oil. One was from the Macondo spill, from the Horizon spill, and the other was a mixture that was sent to us by USGS from 3 different Texas oil fields. So we actually had 2 different sources for our crude oil that we used in these bioassays. We added those at 10, 50, and 100 microliters per liter, or parts per million. And of course, we had a control where we didn't add any oil because we wanted to compare our additions to unexposed communities. So that gave us a total of 90 incubations over the 2 dates. We added nutrients for a couple of reasons. One is because we wanted to see the potential for growth, given that they were not nutrient limited, so that the results that we were seeing are not just simply due to nutrient utilization. So we added nutrients in excess, enough to stimulate the growth of the phytoplankton, and you'll see that result in just a second. We incubated the samples outside in little corrals that were covered with neutral density screen to reduce the irradiance a little bit, because the samples were floating at the surface of the water. But we incubated the samples for 36 hours. We then filtered the samples on the glass fiber filters, a standard protocol for looking at phytoplankton samples. And then we did an HPLC, or high performance liquid chromatography, analysis of their photosynthetic pigments. And from that, we can derive the group abundances for the different types of phytoplankton that were present in that natural sample. So just sort of a quick overview of the way this works is that you take your water sample, you filter it, and then analyze it by high performance liquid chromatography. And that gives you the concentrations of a variety of different photosynthetic pigments that are found in phytoplankton. The cool thing, the really useful thing, is that many of these pigments are unique to a particular group of phytoplankton. And so it's a chemotaxonomic marker that allows us to go in and determine relative abundances of these different groups of phytoplankton. The alternative way is to sit down and look through a microscope and spend hours and hours and only be able to identify a portion of what you actually see. So once we have the pigment data, we can use another program. It's a piece of software called ChemTax. And what ChemTax does is to give you algal group abundances in units of chlorophyll A. So the way to think about it is that the phytoplankton community is represented by all the chlorophyll A. But what ChemTax will tell you is what proportion of that total chlorophyll A is tied up or can be assigned to things like dinoflagellates or diatoms or cyanobacteria. The program does have a little bit of fairy dust and some unicorn horn. It's not ideal, but it's a useful tool, and it's one tool that we can use that can give us some good, very good quantitative estimates of the relative abundances of the different algal groups. So we have a common unit that we can talk about abundances for the different algal groups. So if you look at the initial communities that we started with, the different groups of phytoplankton, microalgae are listed along the bottom, and the chlorophyll A concentrations are along the y-axis. You notice concentrations are pretty low. Typically in North Inlet in the summertime, we get numbers of like 5 to 10 micrograms per liter. And here we're talking about 0.5 to, well, about 1.5 to 2 micrograms per liter when you add up all of the groups. But the thing to notice is that the most abundant groups in these samples were presentophytes and diatoms, with other groups there present in smaller quantities, and cyanobacteria a little bit more abundant in the March samples than the February samples. The point of the slide is to illustrate that it was a diverse community. It is a diverse community. And we're able to know what the starting composition of the community was before the experiments. So if we look at just total chlorophyll A, the common unit that people use to talk about phytoplankton abundance, if you compare the incubation results for the Louisiana and the Texas crude, this is our initial concentrations, and this is after the 36-hour incubation for the control with the nutrient additions, and they grew very well over 36 hours, a doubling or almost a tripling in the warmer waters in March. But if you compare the responses of all of the other groups, there was still not necessarily a huge change from what the initial concentrations were, but they're much lower than they should have been given the amount of nutrients that we added to those samples. And as you increase the concentration of the crude oil, the response declines for the phytoplankton community. And so the conclusion from this is that it doesn't kill everybody, but they don't grow as well as they could under the same conditions if there was no oil exposure. A very busy graph, and I'll explain this in the next slide. I just wanted to illustrate that we can go in and do that same sort of comparison for all the different groups of algae. And I'm not going to go through each one of these graphs. The units here are in the percent mean for the relative response, and the big responders were diatoms and presentophytes. You see there with 50 and 60% of what the initial concentrations were. But just to summarize all of that, the diatoms basically maintained or increased their relative abundance. The chlorophytes and cyanobacteria had a moderate increase, not as high as the control, but they were not inhibited totally from growth. They were still able to grow, just not at as high a rate as they were in the controls. The cryptophytes and dinoflagellates had normal growth at low levels, but as the crude oil concentration increased, we had inhibition at the higher levels. And then euglenophytes and chlorophytes actually showed a modest amount of growth, but not as high as the control conditions. And in the presentophytes, there was really no significant difference from the control, but in March, there was a very clear inhibition of presentophytes with the oil additions. So we can take this a step further and ask really the interesting question for me is, does community composition change? And you can do that using discriminant analysis. It's a pretty simple and straightforward statistical method that allows you to examine all of the responses, the collective responses of the entire community, all the groups within that community. It lets you separate or discriminate groups based on independent variables. In this case, the independent variables were the abundances of all of the different algal groups. And what the analysis will do is it groups similar samples together, so that it's sort of like a cluster analysis where similar samples have the same location in XY space as the similar samples all grouped together in XY space. and it allows you to visualize if there are differences in community composition. So the big question is, does oil exposure result in changes in phytoplankton community structure? So these are what I like to call the sublethal effects. It doesn't kill everybody, but it does have a very marked effect on what the community looks like. So if we look at our functions, function 1 and function 2 for the discriminant analysis, you can see that the control group clusters together, and then over to the right, we see that our 3 separate concentrations are all clustered around the same function 1 axis, but are discriminated by the second function. And what this second function is mostly taking into account the responses of the diatoms and presentophytes to the different exposure levels. The point of this is that exposure does result in communities that are different, and the degree of exposure has an effect on community composition. You can do the same. So the function 1 explains 80%, function 2 is about 16%, and the analysis was pretty good, correctly classifying about 85% of the samples. Another question you can ask is, was there a difference between the 2 oil types in terms of their impacts on phytoplankton communities? So the control, clearly oil has an effect, no oil versus oil. And then the Louisiana crude had a different effect from the Texas crude on the phytoplankton community composition. So function 1 explains about 76, function 2 explains about 23, and about 100% or 100% of the samples were correctly classified. So a very good analysis. I'm very confident that there is a difference in phytoplankton responses to the 2 different oil types. So the additions of crude oil at all concentrations resulted in a shift in the phytoplankton community composition. And the community responses were different for the 2 different crude oil sources. So what do we conclude from this? Well, as you increase the exposure concentrations to phytoplankton, of phytoplankton to crude oil, you're going to see a reduction in phytoplankton biomass. Not necessarily a complete annihilation of phytoplankton, but they don't grow as well as they do when they're not exposed to oil. Algal group responses can range from positive. Some do a little bit better when they're exposed to oil. Some do a lot worse, and you can have negative growth or reductions in the abundances. Crude oil exposure at any concentration is going to shift the phytoplankton community composition. And those phytoplankton responses are going to depend on the community that you start with. So it depends on the community composition at the time of oil exposure. And the bigger picture perspective of this is that you're changing the diet for all of the grazers in your system. Things that depend on phytoplankton for food sources are seeing a different food source than they would normally see. So that has big implications for the trophodynamics of these systems and also for biogeochemical cycling, because different algae do different things in terms of the biogeochemistry and biogeochemical cycling in the water column. So I think the take-home message I would like for most people to have is that phytoplankton responses to oil exposure depend on, pretty much everything, okay? There is no singular common response of phytoplankton. There are a lot of different factors that are important for determining how those phytoplankton are going to respond. But I think the one thing we can say is that even low concentrations of oil exposure can have these sublethal effects on community composition. And forecasting how a phytoplankton community is going to respond to oil exposure is almost impossible. We can't, you know, even if you tell me who is there and give me all of these other variables, it would still be a huge guess on my part as to how those phytoplankton community is going to respond. So I think it's safe to say that community composition is going to change, and that can have some potential cascading impacts on the higher trophic levels in these systems. And so I'll end with a picture of my backyard, the North Inlet Estuary, and it's a beautiful place. If you're ever in South Carolina, you need to look me up and we'll give you the red carpet treatment. So with that, I think I'm just about on time.